Hello and welcome to this Technology Networks webinar, using mass photometry to power up the development of AAV-based therapeutics. I'm Molly Campbell, Senior Science Writer at Technology Networks, and I'm the moderator for today's event. We have a fantastic presenter for today's session, Dr. Matt Rannigan, who will be sharing valuable insights with us. Matt is a Senior Applications Scientist in the Product Marketing Management Group at Refine. He has over 10 years of industry experience in small molecule drug discovery and developing gene editing technologies. As a protein scientist, he has worked with various classes of proteins, including GPCRs, enzymes, multimeric complexes, and ribonucleoprotein complexes. Matt holds a PhD in biochemistry from the University of Connecticut, where he studied the application of photoactive proteins in biophotonic devices. Following the presentation, we'll have a Q&A session where you can raise any questions that you may have for Matt. To submit your questions, simply type them into the box on the right-hand side of your screen and click send. We'll do our very best to address as many questions as possible during the time we have available today. If you encounter any technical issues during the webinar, please click the chat box on the right-hand side of your screen for assistance. And now, without further ado, I'm delighted to hand over to our esteemed speaker, Dr. Matt Rannigan.
Thank you. So my name is Matt Rannigan. I'm a Senior Application Scientist here at Refine. And today I'll be talking about the application of mass photometry to power up the development of AAV-based therapeutics. Production of recombinant AAVs takes several weeks and requires multiple purification steps after harvest. This process results in the purification of the full AAV product, which contains the properly packaged single-stranded DNA for the AAV and the inserted transgene cargo. Several AAV-related impurities co-purify with the full product and can be challenging to remove during production, as many analytical methods are left to the end because they are too slow for in-process monitoring. These primarily include things such as the empty AAV shown here in the center, the partially full AAV, or the full AAV product shown as cartoons on the bottom. While genetically inactive, because they lack a complete transgene or have no DNA cargo at all, many of the AAV impurities pose risks because the viral material can still be recognized by the patient's immune system. In fact, many processes produce far more empty particles than full ones. The risks from these impurities include low transgene efficiency due to receptor binding competition, increased immune response from high viral titers, limited target genome concentration due to capsid precipitation, increased cost of goods produced. A major reason for the high amount of empty and partially full particles, even at the manufacturing level, is the lack of a technology to rapidly and easily determine the empty-full ratio, specifically during early process development. This table from Zhang and Dalby at the University College of London lists the current analytical methods and respective challenges for the different stages of AAV production. There's a very real technology gap for the quick assessment of the relative empty-full ratio of AAVs. Many of the methods listed here are well known to be time-consuming, require advanced training or expertise, or not quantitative, among other challenges listed in this table. Quantification of the full AAV product purity are often left to the end of the production as a result. We strongly believe that mass photometry can fill the technology gap for fast and easy assessment of the AAV empty-full ratio. To explain this further, let's start by looking into how we measure the weight of molecules using light. We measure AAV particles in solution by looking at their light scattering properties when a particle lands at the glass interface. Every single particle leads to one specific dot, giving us single molecule resolution that is represented as counts in the histogram shown here at the bottom. Interestingly, the contrast of the patterns, as in how dark the center of the dots are, is dependent on the scattering of the particles as it lands, whereby more scattering, the bigger and optically denser the particle is. Both of these parameters determine the weight of the molecules, and hence by analyzing the contrast of the single molecule events, we can determine their weight using a simple contrast to molecular weight calibration. The output of mass photometry is the histogram shown at the bottom of the figure, showing the contrast and hence the molecular weight for every detected particle counted by the instrument. Now, measuring the weight of particles is very useful to analyze the empty-full ratio of AAVs due to the significant weight difference of about a megadalton between the empty and full AAV particles due to the added weight of the DNA. Also note that low molecular weight contaminants will not interfere with the measurement, as they are typically much lighter than the assembled virus. A typical mass photometry histogram is shown here for a mixture of empty and full AAV particles, with a nice baseline separation between the two populations, shown here between the two populations. And the empty and full ratio can easily be determined by the software through counting the molecules in each respective peak. Each measurement takes only minutes, does not require a trained operator, consumes very little sample, and can be measured in native conditions with little sample preparation. Here we see some examples of some of the applications for measuring empty-full ratios of AAVs. In the left-hand figure, we show the empty-full ratio has a linear correlation for different AAV loadings from 0 to 95%. For this demonstration, we had purchased and analyzed empty and full AAVs separately or mixed at different calculated ratios for 5 different data points across the entire range. The linear correlation is shown as the inset figure in the upper portion of the left-hand figure. The center and right plots respectively show examples where we observe counts for partially filled capsids between the two peaks and overfull capsids outside of the gated regions for the empty and full particles. The presence of these partially and overfilled species has been confirmed using orthogonal techniques like sedimentation velocity analytical ultracentrifugation, and we'll be showing literature examples in the coming slides of how these two methods compare. AAV analysis with the SamuxMP is serotype independent, as shown in the left-hand figure for 4 different AAV serotypes, being AAV5, 6, 8, and DJ. These samples were all measured with the same experimental routine, and we did not observe any specificity for certain serotypes or performance differences. Higher order AAV aggregates can also be detected with the SamuxMP, as shown in the right-hand image. In this example, we induced aggregation for AAV8 by drying and reconstituting the virus. We then measured the sample and calculated the mass ranges for multiple AAVs, shown in the light blue regions of the right-hand figure. The population at the mass range for 2 AAVs was significant enough to fit a Gaussian. At bigger AAV aggregates, we could detect counts in the calculated areas, but the populations were not large enough for us to fit a Gaussian and test for linearity of the system. Hence, we conclude that the SamuxMP can detect aggregation. However, quantification of those aggregates has not yet been tested and validated fully. Another application for the Samux mass photometer is that it can also be used to estimate capsid titer of AAVs within the right order of magnitude. Tested has primarily been limited to purified AAVs thus far, and samples must be diluted within the mass photometry concentration range of about 10 to the 11 particles per ml. What we can see in the left-hand figure is that for the test cases represented in this figure, mass photometry, shown as the blue chart bars, performs and estimates AAV titers that are comparable to those determined by traditional ELISA methodologies. In the example on the right, Pharmaron has also shown that this system can aid the optimization of process development protocols. Analyzed samples were collected at different stages in downstream purification processes, from post-harvest clarification through chromatographic column separation. Analyzing column elutions shows that the empty AAVs are found in all of the eluted fractions. But elution 4 is the most enriched for the full AAV capsids in this particular sample. The key is that informed decisions can be made quickly with the SamuxMP using only a small amount of sample for either capsid titers or in-process sample identification. The SamuxMP can also be used for GMP regulated environments, as we offer software that's 21 CFR 11 compliant for easy data integrity and traceability, with features such as user authentication, access control, audit trails, and electronic signatures at export. The system also has qualification and support for easy system validation, including comprehensive training and documentation, and IQ/OQ. It's also worth noting that mass photometry is now listed as in the British Pharmacopoeia for regulatory guidance of the production of recombinant AAV particles. Mass photometry is highlighted in the center with the blue rectangle, and the gold standard for this is analytical ultracentrifugation, shown in this column over here. And these are listed among other techniques such as anion exchange chromatography, charge detection mass spectrometry, transmission electron microscopy, and then size exclusion, SECMALs on the very right-hand side. Mass photometry, as this table shows, requires small sample volumes. The time to result is less than a day, with a single measurement and analysis typically being at or under 5 minutes, which is faster than most of these techniques, which require a full day for both. Requires low expertise relative to these other techniques. Is also listed as high value for suitability for in-process control methodologies, and is noted to have fully resolved intermediate and overfilled resolution for these AAV particles. There are two versions of the SamuxMP mass photometer that are available, one manual and one automated system, whereby the SamuxMP Auto allows the user to run and analyze up to 24 samples without operator intervention. Here, the auto robot performs the pipetting steps for each sample, which reduces potential error from a human operator while allowing the user up to an hour and a half to focus on other tasks. And so we'll turn our attention now to how mass photometry is being used for AAV characterization in the literature for some real-world examples. In this slide, there are three reports of linear correlation for the empty-full ratios presented earlier using mass photometry. These reports are from different labs, both in industry and academics, and you see that all of the samples have a very good linear correlation of greater than 0.99 for the R-squared value. You'll also notice that each example uses a different serotype, demonstrating that mass photometry can be used for multiple serotypes, again, supporting the serotype independence of the technique. And on the right-hand side, mass photometry, shown as red triangles, was compared against ultracentrifugation, fluorescent ELISA technique, and PCR ELISA, and you can see how it performs, as well as the AUC technique there, with the ELISA PCR sample with purple X's deviating the most from that trend. In another example from one of those groups, they showed how the SAMX performs consistently with the addition of excipients that are common to the AAV field. The figure on the left-hand side shows measures the background signal as a root mean square deviation, or RMSE value, for the acquired microscopy image. This value should be as low as possible to not raise the lower detection limit of the instrument. And you can see here that the presence of the tested excipients only slightly increased the background signal near the accepted criterion range of 0.07, represented by the blue bar in the inset, with exception to a few additives, and the greatest exceptions being the two on the right-hand side for polysorbate 80 and polyvinyl pyrrolidine, which are well above that threshold. The figure on the right-hand side shows how the buffer component being with AAVs being measured either just in water or phosphate buffer with various levels of salt can influence the number of counts that one can get on the system. However, the empty full ratio represented by the triangles and the diamonds in the figure remains relatively unchanged for all these conditions. In another example from the literature, there was a paper in 2023 and 2024 that looked at the empty full ratio analysis of mass photometry for AAV9 and defined the four regions of the four mass regions for the mass photometry measurements, from left to right being the empty particles, partially full, full, and overfull particles, and compared this specifically to sedimentation velocity analytical ultrasonification, or AUC, which again is the current gold standard for empty full analyses. And you can see for AAV9, mass photometry, shown as the purple bars, performed as well as the AUC in this report from Wagner et al. That paper also looked at other AAV serotypes, again supporting that the technique is serotype independent. You can see on the left-hand side and the center that they also looked at AAV5 and AAV8, where the same color coding is in place, and the respective estimates for the different populations is comparable to AUC in these cases. A different paper in 2022 did the performed empty full analysis for AAV2 and AAV8 and compared mass photometry against CDMS. And so what we see on the left and right-hand side are those analyses of the two methods for AAV2 and AAV8. And from the top down, the authors looked at three AAVs from three commercial sources, being Virovec, Sirion, or Vigene, and quantified the relative amount of empty, partial, full, and overfull particles. And what you can see from these figures is that mass photometry performed as well or better than the CDMS in estimating the resolution of these different populations. Again, with the added benefit of fast sample time and low experimental development. Another paper from Richter et al. In 2023 looked at a comparison of multiple methodologies for analyzing empty full ratios of AAV particles, whereby the author's impetus was to compare analytical ultrasonification to the other leading instruments for those analytics, and with the caveat that analysis of partially full AAVs was only reported for analytical ultrasonification. And for most of the cases, all of the partials were less than 4.5%. There was one case for AAV9, which I'll talk about in a minute, where those partials were at 8.1%, and is notable. But starting on the left-hand side of the slide, for AAV2, we see that the four techniques used in this for this figure were mass photometry, AUC, transmission electron microscopy, and anion exchange chromatography, shown in dark orange. And what we see here is that there are three samples measured for empty full analyses of AAV2 in this case. There was the sample that was labeled as empty, with good agreement among the four techniques for being containing primarily empty particles, full AAV2 particles, and then in-process samples on the right-hand side of this figure. What we can see is that in all of these, mass photometry and AUC are effectively equivalent in their estimations of empty full analytics. TEM, shown here in light orange for the full particles, seems to over and underestimate those ratios that ratio respectively, and the AEX technique under and over represents the amount of full AAV2 respectively. The in-process samples appear to be equivalent for these 4 techniques for AAV2. For AAV8, however, we see that, again, the analysis of empty particles for the empty sample is underrepresented by TEM. The measurements for the full particles, again, TEM is deviated the most from these, and the in-process samples have the same story. For AAV9 on the right, it's a similar analysis where MP performs as well as AUC. For all of the samples, TEM seems to work fairly well for this serotype, and the in-process full sample here on the very right-hand side for AUC, you'll notice is deviated from both MP and TEM. This is that 8, whereby they're reporting the amount of full capsids, and the differential between AUC and these other 2 techniques here is about 8%. And so this is the one case where they're reporting partials for only AUC seems to deviate from the other techniques in a way that may be misleading if the detail's not caught. But otherwise, the takeaway is that mass photometry is consistent with AUC for these 3 different serotypes. And I should point out that for AAV9, the AEX method was unable to separate the empty full species. And so the asterisk indicates that there was no data reported for that technique and AAV9. The same paper also did a comparison of mass photometry for these 3 serotypes, empty full analysis for mass photometry, AUC, and another light scattering technique called the stutter. And you can see here that, again, mass photometry is consistent with AUC, and the stutter is over and under representing those ratios accordingly, shown in orange. In a report from Wagner et al. In 2024, we see that these authors demonstrate a process whereby the schematic on the left-hand side demonstrates how AAV can be cleaned up from harvest material using nanobodies covalently attached to polystyrene beads and pulled out of solution and then eluted from the beads after a cleanup step for empty full analysis. They did this, they ran this experiment for 2 different transfection mixtures represented in the bar chart on the right as the blue bars or the red bars respectively, and 2 analytical techniques being mass photometry as the darker color of the 2, or AUC as the lighter color of the 2. And again, for the different populations of empty, partially full, full, and overfull AAV particles, you can see that the 2 methods performed equivalently well for these 2 different transfection mixtures. In this slide, we're presenting data from a paper whereby the authors used mass photometry to analyze samples purified by size exclusion chromatography, or SEC. SEC traditionally pairs very well with detectors for UV refractive index or multi-angle light scattering, or MALS. However, the analysis of these particles can be a bit challenging for proper quantification, as the presence of heterogeneous genome packing like partials and overfills can skew the data towards one or can skew the empty full ratio data. As you can see here, the samples are effectively in one peak, as the different morphologies of empty, partial, full, and overfull are all of equivalent sizes, and the difference is in the cargo that they internalize. So what the authors did is they took some purified AAVs, mixed them at a known ratio for empty and full, injected over size exclusion chromatography, and then used mass photometry to analyze the relative empty full ratio of the population within the boxed peak on the chromatogram at the top. And what they found was that there was good agreement between the pre and post sample, meaning that they were capturing, none of the sample was lost to the column, and they were seeing effectively a very quick analysis to confirm the percent full in this case of AAVs over the chromatogram. There are also reports for being able to approximate transgene size using mass photometry. In this particular paper, the authors compared 3 different approaches for estimating the size of the transgene, whereby approach 1 used intact AAVs and made the assumption that the contrast differences observed for the different particles are attributed to encapsulated genome of empty and full AAVs. They did use and compare different standards, including protein reagents and DNA or RNA ladders to generate their calibration curves. In method 2, they used intact AAVs whereby for the calibration, whereby the different capsids had either no genome as the empty particle, or a different or a cargo with a differing transgene length from small, medium, and large to get a 4-point calibration. These were known genome lengths whereby they could control and make a calibration curve. And then the third approach, what they ended up doing was what they termed genome release, purifying out the purified genome and then comparing it against standards that were a calibration curve, again, that was produced using either an RNA or single-stranded DNA plasmids. The paper goes on to present tables and allow statistical analysis. And so I'll just summarize that by saying they conclude that approach 2 was the most accurate, and interestingly was their replication of another paper from the literature, indicated by the asterisks in the top. And here they saw the most accurate results for estimating transgene size. Approach 1 gave a general overestimation of the transgene size by up to 5%. And the genome release method, they noted, was accurate with an RNA calibration, but had a much lower resolution due to the presence of aggregates fragments from the purification process of the single-stranded DNA after release. Transgene size was also reported in a 2023 paper from Wagner and colleagues, whereby they took 3 different samples of AAV9, did their empty full analysis, and then calculated the transgene size based on those the differential mass, and compared it to the calculated size from the known number of kilobases in the transgene. There was a relatively good agreement between the calculated and observed transgene lengths. They did note that the longest transgene had the highest error, and in looking at their methods, this is likely attributed to their calibration curve, whereby they used an empty AAV9 and thyroglobin, which is a protein standard, which again, like the longer the transgene, the higher the error rate for the way that they were standardizing in this paper. And that is to say that if they were to have used approach 2 from the previous paper, it'd be interesting to see what their data report looked like there. They do conclude, however, that mass photometry is faster than other methods for empty full analysis and transgene estimation, like SecMols, and requires no prior knowledge of the analyte, such as the extinction coefficient for the nucleic acids. A 2023 paper from Wagner and colleagues shows how mass photometry can be used to estimate capsid titers. And again, this is, you'll notice on the figure on the left that there's a good linear correlation between the binding count rate for mass photometry and the capsid titer determined by ELISA with an R-square of 0.99, greater than 0.99, for the range of 0.4 to 8 times 10 to the 11th capsids per mil. What happens outside of this range, as shown on the right-hand side, is that the linearity of the curve diminishes, primarily due to a loss of the MP signal as the field of view for the instrument becomes overcrowded and your counts become unreliable. And this can be observed through the lower counts at higher higher concentrations of the virus particle. You can see that the mass photometry peak diminishes just due to crowding and the instrument not being able to get accurate counts. Other applications that are that seem to be becoming more popular in the literature as of late are stress and degradation testing that can be done with mass photometry. Such examples include thermal stress, pH stress, number of freeze thaws, oxidation, UV exposure, mechanical shear stress, or lifetime studies, whereby a sample is stored at 4 degrees or ambient for a period of time with periodic measurements. The paper cited in this slide demonstrates how thermal stress can be measured by mass photometry. These authors used AAV8 and heated the samples, measured the empty full ratios for samples that were heated at different temperatures for a set period of time. And you can see here that the estimated empty full goes from 80% down to about 40% at those different temperatures. In the lower right-hand side of the figure, of the slide, we see a figure showing their mass photometry data at 3 temperatures, being 24, 54, or 59 degrees, and how they were able to identify 3 species by mass photometry, being the empty population, and then 2 full populations with either 1 or 2 transgenes that agreed well with their calculated values for what the transgene should be. And in all cases at higher temperature, we see loss of both species over time and production of the empty virus as the transgene is presumably released due to thermal stress. In another example from a different research group, we see how they used thermal stress to take their AAV8 sample, and after an initial reading at room temperature, subjected the sample to 65 degrees for 15 minutes, saw an increase in the population of empty virus, and what they determined to be a population of the transgene down at about 0.7 megadaltons, which they concluded was a release of the single-stranded DNA. They determined this, one, because it was about the right mass for what they calculated, but also because after taking those heated samples, they did a second incubation for 15 minutes at 37 degrees with DNase and saw that population disappear, supporting the identification of that population as DNA, and that's subject to hydrolysis. A secondary observation was that their population of full AAV diminished to a point that was notable, producing more of the empty viral capsid in the presence of DNase. These data are represented again in figure B in the top right-hand side of the slide, whereby you can see comparison of the heated samples with and without DNase and a proportional loss of the peak for the full capsid and that transgene size at about 0.7 megadaltons. They quantified the percent full in the bar chart on the right-hand side of figure B as 50% without DNase and about 30% with DNase, and came to and concluded that what what they figured was happening was that not only were they heat denaturing the AAV8 serotype samples, causing release of the genome in some population, but because they had a loss of full particles with the addition of DNase, they must also have a population whereby those AAV particles hadn't fully released, but still had the DNA exposed in a way that the DNase enzyme found accessible, right? And so it was it found accessible and led to a further population of loss of their mature full AAV product. In figure C, in the lower right-hand side, they represent these data again, but now as a change in percent filled from left to right as a function of temperature, and show their replicate values as a function of count for the amount of AAVs that they're able to identify in the mass photometry figures as a function of the size of the circle. This paper also shows how these stress tests after heating with DNase can be done for different AAV serotypes and may in fact be particular to different serotypes, where again, figure A and B are the data from the previous example for AAV8 plus and minus, whereby we see, again, after heating and with the introduction of DNase, we see loss of the full viral capsid. AAV2 does not seem to have that same subjectivity or effect when heated and or exposed to DNase, showing that AAV2, at least for this these samples reported by this group, AAV2 didn't show the same thermal stress or DNase sensitivity that was shown by AAV8. And so with that, then I will summarize the benefits of mass photometry and the SamuxMP in particular for working with AAV characterization, production, and purifications. I hope that I'm able to convince you that the SamuxMP's measurements and performance does make it a great tool to have for anyone working with AAVs. But notably, apart from the measurement performance, there are more reasons that make this technology useful for AAV characterization, including limited sample preparation, as this is a label-free technique that measures particles in their native state. The results are fast, as it takes less than 5 minutes per measurement, including the analysis, requires low sample amounts, typically 10 to 20 microliters of around 10 to the 11th particles per ml. It's simple to use. Training of new operators can be done in less than a day. The instrument has a small footprint and fits comfortably on a benchtop, and has low operational costs, as is less than $5 per measurement. And with that, I will leave here for a second the summary of overall applications presented in this webinar and highlight that the details that we covered were that mass photometry has been adopted as a regulatory instrument according to the British Pharmacopoeia. We demonstrated literature examples whereby the authors were able to analyze empty, partially full, full, and overfull AAV particles. There were applications for upstream and downstream in-process monitoring of AAVs. Authors demonstrated that they were able to estimate the size of the transgene from empty full analysis with proper calibration. They were also able to estimate capsid titer quickly and reliably compared to ELISAs. There were the 2 examples at the end where authors showed how one can use mass photometry for quality control and or degradation testing of their particular samples. And with that, I'd like to take any questions, and thank you all for your attention today.